Dr. Damaris works closely with other disciplines from national, private, and international institutes to develop cutting-edge genomic resources for all ICSAT mandate crops. And those are sorghum, ball and finger millets, groundnuts, chicken pea, and pigeon pea. Prior to this, she worked as a senior researcher at the Agricultural Research Council, ARC, in South Africa, where she implemented molecular breedings for several indigenous crops, including sorghum, cassava, sweet potato, African eggplant, and amaranth. Dr. Damaris is also passionate about mentoring of young, upcoming scientists and has successfully mentored more than 50 scientists in her career today. She leads the Science and Publication Subcommittee of the Africa Plant Breeders Association. Her area of expertise are qualitative and quantitative genetics, genetic mapping, germplasm characterization, pre-breeding, computational biology, high-throughput genotyping and marker development, next-generation sequencing, and data analysis, transcriptomics, and metagenomic analysis. She's the editorial board member of the Reach for and Applied Genetics, the Plant Genome, Plants, People, Planet. Ladies and gentlemen, allow me to invite Dr. Damaris. Dr. Damaris, take the floor.
Thank you. Thank you so much, Wilfred. Can you hear me? Can everyone hear me?
Yes.
I have two screens with me, and I'm not too sure which screen you're seeing. Is it the one that has the full slide or the one that's split? Otherwise, I'll switch it.
We are seeing the one with the full slide.
Okay. So yeah, thank you so much for the very, very kind words for that nice introduction. And also thank you for considering having me in this seminar. As I mentioned, also you did mention in my introduction, I'm very passionate about mentoring students. So I always will go for such invitations, especially when PhD students are involved or students generally are involved, just to share the little that I know and also to network. Most of it is to really network with everyone. So thank you for this invitation. I'll start. I believe I have 40 minutes, so I'll try to use about 25 minutes for the presentation and 10, 15 minutes for questions and answers. So my broad topic is basically on using genomics to enhance productivity of dryland crops. I will mostly work on finger millet, and you'll see why. Okay. And just to start by saying a little bit about where I work, which is at the International Crops Research Institute for the Semi-Arid Tropics. You can get a little bit more on that website, but we work on 6 main mandate crops, 3 cereals and 3 legumes. We also work on small millets, but these are the main crops. And the main characteristic of the crops we work on are that they are able to thrive in the semi-arid tropics, usually in places where you get very erratic rainfall. Farmers are relatively poor, so they are not able to afford irrigation. And so most of the crops that we work with are both for subsistence, but if they have surplus, they can sell them to the markets. And we look both into the nutritional value and also the quantity of yield. Most of our crops respond to these 6 strategic development goals in terms of no poverty, zero hunger, good health and well-being, responsible consumption and production, climate action, life on land, partnerships for the goals. So what are dryland crops, one would ask? Yeah, I mean, it's very simple, just like the title says, they are crops that are able to survive and produce under dry conditions. And I just put on the screen some of the crops. Most of them are the ones that I work with. But there's quite a bit of attention now that is being given to dryland crops for obvious reasons. Most of it is climate change. But especially in the African context, even before we start talking about climate change, we have not much irrigation. So most of the farming that we're doing is really dryland farming. And so there's very much interest in dryland crops. And most of these are crops that are native to our continent, or, they've been grown there for a long time. And one in which I'm going to focus today, as I mentioned, is finger millet. For those of you who haven't seen finger millet, this is how it looks in the field. And part of the reason why I really wanted to talk about finger millet is because finger millet is mostly an East African crop. So in West Africa, it's almost not known, but it's gaining importance. People are becoming aware of it. And I thought, since this is a West African symposium, maybe enhance the awareness on finger millet in this forum. And just to say a little bit about finger millet, it's mostly, first of all, apologies, I couldn't find a better map that has the South Sudan. So if there are any South Sudanese here, no offense, I just wasn't able to find a better map that has these production areas for finger millet. But finger millet is thought to have been domesticated almost 5,000 years ago. It's actually being thought to be one of the oldest domesticated cereals in Africa. And the domestication is thought to have been around Ethiopia or Ugandan highlands. There have been theories of different domestication events, but there hasn't been enough studies to actually confirm some of those theories. And so for now, we continue to study to really understand where domestication actually happened and whether there were parallel domestication events. And the likely areas of domestication would be Ethiopia, Uganda, and perhaps South Sudan. And that's because these three regions have very broad genetic diversity for the crop, including wild relatives. But also they have very distinct types of finger millet that are known to have been grown over the years for a long time. And so, yeah, a lot of studies still need to be done to debunk that theory. It is a tetraploid. One of the genome donors is known. The second one is not known, and it's thought to have been extinct. But to be honest, it's too early to tell. There's very little that has been done to specifically look for this elusive second genome donor. And so maybe, who knows, with more characterization, we may find it. But I also put on this slide, we do know that there has been movement of finger millet to West Africa, and there's a particular region of interest in Nigeria, in the Taraba, Plateau, and Kaduna states, where finger millet has been grown for years. This finger millet is thought to have been introduced there from different parts of East Africa by some of the immigrants. But now it's actually quite significantly grown there, and it is expanding. We also do, as ICRISAT, we have planned releases in Ghana, Mali, Niger, and Burkina Faso. And most of the West African germplasm remains uncharacterized. The most important attributes of finger millet is actually its nutritional quality. And for its nutritional quality, it actually fetches way more in markets. And this is more or less globally, including in Eastern Africa, where it is mostly produced. And one of the most important mineral composition or nutrient content is its calcium content, which is almost 10 times as much as you find in the major cereals, wheat, rice, or maize. But beyond that, we also know that finger millet is really superior in terms of its malting quality, it's gluten-free, It's adapted to diverse agroecologies. It has very good grain storage quality, and that's because of the high polyphenol content. And so you don't get a lot of storage pests. So you can store it for years. I've actually, so I started working on finger millet 11 years ago now. And what I did at the time, I was just curious, and I took a bag of finger millet, which I've kept at home. The last 11 years, it's still intact. And I don't even, it was just in a small envelope. It's still intact. I don't keep it really well. It's just in one of the drawers in the kitchen, but nothing has infested it so far. So it's pretty good. But finger millet is also used as stover for fodder. The grain is also good as feed. And especially in East Africa, it is really the cereal to go for as weaning food, and also for patients. So most of the time, this is what will be given to patients. Now, it is a good source of income because the demand outstrips supply. It's almost every year there's huge demand for finger millet that the subsistence farmers, the predominantly subsistence farmers, are not able to fulfill. So this makes finger millet quite an interesting crop to work with. And our major points of intervention have been mainly 4 areas. That's in germplasm or pathogen characterization, then doing trait discovery, validation, and integration, and then attempting to implement genomic prediction selection in finger millet. And we also hope that we can go all the way to varietal release, adoption tracking using genomics tools. Now, all these activities we implement through capacity building. So capacity building is a really important part of all the activities we do to ensure that, at the end of the day, the capacity that we need to sustain the research in finger millet is retained in the continent, especially. Just a few years ago, working together with collaborators from Scottish Rural College, we did a study where we were looking at the distribution of finger millet, all species in Africa. And this work is already published by Banchich, that this was published last year. It was interesting to see, so the data that we used here was obtained from Global Biodiversity Information Facility, but it was interesting to see, we have this broad diversification of finger millet that we can find in public databases. But I mean, clearly there was absence of that diversification or that material for Ethiopia, which is surprising because Ethiopia, as I mentioned, is one of the centers of domestication. And we do know that Ethiopia does have significant diversity that needs to be characterized. Now, I mean, if you see, I mean, these are the number of accessions used. So Ethiopia is actually one of the, in this database, you have very little from Ethiopia, which is quite concerning. But in a different study, we also decided, just specifically to characterize Ethiopian and Ugandan material, and we got really interesting dynamics, as you can see here. So we see distinct clustering of Ethiopian versus Ugandan material, which in a way might suggest the distinct or parallel domestication events. But if we look at a broader germplasm characterization where we include India or Asia, by the way, I didn't mention that finger millet is thought to have been introduced into Asia or India and Nepal about 3,000 years ago during trade. And now finger millet is a very important crop also in India and Nepal. Now, when we do characterization that involves material from India and Nepal, obviously these have been diversified for 3,000 years. We get very distinct clustering, but we also get a little bit distinct characterization or clustering from the South African germplasm. And we do know that most of this germplasm and its diversity is yet to be exploited for crop improvement. And so this is some of the things that get us really curious to look at, okay, what is it that we can find from this diverse germplasm in terms of just understanding what are the novel traits and how we can use it in crop improvement. But again, we are very curious. We've only attempted to characterize quite a few, just a few. Maybe in this study, we characterized about 350. And this pattern of clustering is very interesting. It also means that there's very little exchange of germplasm between the two pools. But for those of you who come from Ethiopia and have seen Ethiopian finger millet, it's very distinct from what is grown in the other part of East Africa, Uganda, Kenya, Tanzania. And so obviously there's need to characterize more to be able to really understand the diversity there and how we can utilize it in breeding programs. Now, I'm going to switch to pathogen characterization, and the work that I'm going to present here is work that have been done by Angeline, who is a PhD student currently at the University of Nairobi and is doing her PhD work under my supervision at ICRISAT. Now, the blast disease is one of the most devastating diseases in finger millet, and it affects leaf, as you can see from these pictures. So you can get these kinds of symptoms, or it can affect the neck. Obviously, once it affects the neck, then the head is not going to be stable and it will just drop. Or it can also affect the finger itself, as you see in these pictures. So up to 90% yield losses can be observed when conditions are favorable. Now, we don't really understand the dynamics of this pathogen. Bear in mind, this blast disease is similar to what we have in rice. And for those of you who are involved in rice pathology work, rice blast is one of the most characterized, most studied. We know individual races and where they affect and the disease, the corresponding resistance genes. Yet in finger millet, we don't even know the dynamics within a particular country, leave alone a region. And so in this particular study, we had collected about 450 symptomatic samples from leaf, neck, and finger across growing regions of Kenya. And then when Angeline started her PhD work, she started to culture them in vitro, and she did morphological, molecular, and pathogenicity characterization of the isolates. Now, it's very interesting. I mean, this is probably one of the few studies where we actually are able to see now the differences in morphological differences and be able to see how we can identify different isolates just by looking at morphological features. And some of the ones you see here, we used it to characterize. Some of them, obviously, when you do, one of the things that is obvious when you're doing pathogen characterization is that sometimes what you culture, especially when you're doing something like fungus, sometimes what you culture is not really what it is. So morphological characterization is not going to be enough. And so the other thing that Angeline did was also to do molecular characterization. Now, we did molecular characterization using ITS and PFH, as well as POT2 genes that were amplified. Now, I've just chosen to highlight here the POT2 clustering, which did a really good job at clustering the Elucine pathotypes, which are the finger millet pathotypes. So now here in this screen, we're actually seeing potentially 8 pathotypes, but obviously we need to be able to do pathogenicity tests to be able to really distinguish them as different pathotypes. But we do know that these are clustering together. This one cluster is probably likely to be the same, and then we have a little bit of differentiation shown in these ones here. And so what Angeline also did was And I mean, just to say that you can see clearly, this clustering is very distinct from the rice pathotypes or wheat pathotypes, which is pretty good. But the other thing that Angeline also did was to be able to select two isolates. So she selected 72, this particular one, and this particular one to be able to do some pathogenicity tests. Now, she had to also identify or select a differential set. And so she used different regions and genetic diversity to select some different genotypes for doing the pathogenicity analysis. And while there's not so much difference in terms of their response to S2 and S72, we find a case where in this particular genotype, ICFV 182512 is susceptible to S2 and appears to be resistant to S72. And these were studies that were done in vitro, obviously will need to be replicated in the field so that we get to a point where we actually know that distinct pathotypes are probably responding to different genotypes differently. And that also would allow us to be able to identify the particular corresponding resistance genes. Now, remember, we saw, sorry, I want to go back. We have different clustering here. We've only done, so Angeline only did this 1 and 2. So we still have quite a few to be able to do. And also just to say, this cluster here of port 2 is only having a few distinct ones. We ever think about 47 that appeared distinct. And so a lot more studies need to be done. And so, you know, I mean, especially for PhD students, if, you know, someone would be looking for a PhD project, it forms a really nice study to be able to characterize all of them, to be able to identify what are the likely pathotypes. Now, moving on slightly, just now to move to trait characterization or trait discovery, we typically use, and I mean, this is standard, we typically use, just like in any other studies, association mapping and biparental populations for trait discovery. And so in this slide, I'm showing what we typically would use. So in sorghum, we use reference sets like global reference set or mini-core set or association panels. In finger millet, we have a core set that has been developed. It's not really as optimum as this is, and there's a reason why I'm saying that, and you'll see why I'm saying that towards the end of this presentation. But any good diverse set would make for a nice association mapping population. But as we have done in other crops, it would be nice also to do an association mapping panel or to characterize one that can be used for finger millet that is exhaustive and is representative of the diversity that exists there. But again, we also do biparentals where we look at genotypes that are contrasting for a specific trait, and then develop mapping populations, and then do trait discovery. Now, this doesn't have to be as simple as it looks, because now we have very advanced tools. We have nested association mapping that we can do using multi-parent populations if we have founders of different traits of interest. So you can project this to, I don't know, 20, 30 multi-parent populations and still use it for trait discovery, as long as you understand the background. But for simplicity, I've just put here, you know, biparental for better understanding of the procedure. Now I'm going to give two good examples that we've done in finger millet, and I'll start with blast disease just for continuity, because I was already talking about it in terms of what we did with Angeline. Now, Emmanuel, who was a student, so now he's more or less done with his MSc from Makerere University in Uganda. did a study on blast disease trait discovery. And just to mention that that study was building on other studies that have been done. We've had a biparental QTL mapping study that was done and has been published already, where we identified leaf blast severity on chromosome 8b as a really good signal. We had other signals as well. But in this particular case, Emmanuel did association mapping, building on a study that had been done previously in Kenya, where we identified quite a number of resistant lines and that were really contrasting with susceptible. So we included some of this in his study, and he also included Ugandan lines, and he screened them across one environment for 2 seasons, and then looked at association with the trait of interest. And this is just to show how diverse the germplasm was. Now, I have to mention that this study was done with ultra low SNP numbers because basically we didn't have funding to generate good SNP data. And so we just capitalized on the data sets that were existing. So we ended up with very low numbers of SNPs, but we were able to filter. So Emmanuel was able to filter those SNPs really, really well and ended up having a set that was quite informative. And one of the things that people don't really understand, and I know especially for students, usually there's this rush to have several SNPs for your trait discovery. You want to have 1 million SNPs for your GWAS analysis. But I've seen even in presentations where people used like 1 million SNPs, but if you look at informativeness of those SNPs, you find they're like explaining 4% of genetic variation, which basically means they're not really that useful for your population. And I'm here to just show that in this particular case, Emmanuel used ultra low numbers of SNPs and just filtered them really, really well. And despite them being really, really low numbers, he was still able to get really good association mapping done in his study. What we do with finger millet, since we have finger, neck, and leaf blast, we always score for all of them because, you know, in some genotypes, they get devastated more at seedling stage or, you know, or by just the neck isolate. In some cases, it's the finger. So he looked at all those. This NBS stands for neck blast severity. This is finger blast incidence, finger blast severity, head blast incidence, and leaf blast severity. And here to just share some of the results. It was interesting. So you can see even the distribution of the markers that they were really low, but he was still able to pick some interesting association. If you see this particular one on 9B, it's actually consistent both for leaf blast severity and finger blast severity. And the distribution of the traits really showed quantitative variation across the years. So it looked like a really good study. So we went ahead and looked at, you know, the ones that looked really promising. And I'm just highlighting here chromosome 9B, for example, which was consistent in these 2 instances. And we also end up with very promising candidate genes for it, which is promising that, you know, this study can be expanded further to be able to implement some of these markers identified in the breeding pipeline. So moving on, the last example that I'm going to give here is also for trait discovery, a study that has been done in Ethiopia. And I tried in this presentation to be able to give studies that have been done across different countries, but also with different traits, and how we are actually trying to coordinate that as ICRISAT, you know, providing support across different countries. So as I mentioned, Ethiopia is actually considered a center of diversity. You know, there are archaeological records showing that finger millet was used dating back really several years ago. And we also know that there's huge morphological and genetic diversity. Now, one of the major things is that we have very little of Ethiopian germplasm represented in global gene banks, which is a concern. I mean, like if the Global Gene Bank is supposed to reflect the exact diversity, then it should really show or reflect, especially in the centers of domestication where we have the highest diversity. Now, unfortunately, in the ICRISAT Gene Bank, which holds the most collection of finger millet, we only have about 1,488, which is about 20% from Ethiopia. And just to remind you, Ethiopia is also a big country. So that's a concern. Now, in this particular study, what Odeny did was, the work I'm presenting here was done by Odeny, who is a PhD student at the University of KwaZulu-Natal, South Africa, but he also works at the Ethiopian Institute for Agricultural Research. Now, what he did in his study was he collected native finger millet from these regions. For those of you who've worked in Ethiopia, you know these are the major finger millet growing regions. But he also analyzed them together with all the improved varieties, like the released varieties in Ethiopia, which was 23. So altogether, he used 448 genotypes. Now he evaluated them across 3 environments. So you see E1, E2, and E3. That's Acinegele, Maitsebri, and Maizo. And he collected data both for agronomic data and nutrient data. Now, for the purpose of this presentation, I'll only present the nutrient related sorry about the spelling here. The nutrient-related traits that we analyzed, and that is for iron and zinc. So one of the things that we started looking at was, okay, so now we know that the finger millet germplasm is really underrepresented in global gene banks. How does that look in terms of the released varieties in Ethiopia? Are they really utilizing all the diversity that is present in the country? And we clustered the germplasm together with released varieties. And we found released varieties clustering together with, now most of the germplasm in this cluster here are actually from SNNP, Southern National Republic, that region in Ethiopia. And so almost 90% of the released varieties clustered here. So where you see the arrows are where all the other released varieties that didn't cluster here are clustering. And so you see certain areas like Tigray, where I've highlighted here, there's almost no released variety that has genetic origin there, which is obviously a concern, but part of the reason why we did this study. So with respect to iron and zinc, which is these two are very important traits, Odeny then just used NIR and looked at the range or distribution of these two elements across the different environments. And different environments varied. E1 probably had the lowest. Usually there's a bit of correlation between iron and zinc. And environment 3 had always the highest. Now, if you look at this range that was recorded by Odeny vis-a-vis what we have that has been reported from the main collections, which are basically from ICRISAT, the range is quite different, very, very different. So you see here iron, the ranges that we see here are very different from what we see in the gene bank. The same with zinc, but zinc is the one that is drastically different because this is the range that has been reported. But here we have up to 8.1, which is almost 4 times what has been recorded in the past. This is great news, but something else that we wanted to see is how are then the released varieties performing in comparison to the land races? Because you'd expect that, the released varieties, they're improved, so they should perform better. Now, I'm not presenting the agronomy work, but with respect to iron and zinc, we looked at the top 10 performers, the top 10 genotypes that had the highest iron and the top 10 having the highest zinc. And I just chose this one location, Arsenigal. And we didn't find any of the improved varieties among the top 10. In fact, in most cases, they were not even among the top 30, not even among the top 40. Clearly indicating that we are not really utilizing the diversity that we have in regions in our countries, and especially in this case in Ethiopia. But specifically, it's ironical that we would find that Tigray material are the superior performing, yet they are the least represented in the released varieties, right? So this is just an example to show us that, there's more that we still need to do with finger millet. And, if there are partnerships that we can form to do a little bit more. And remember, I did mention before that, we still think that the BB genome donor is elusive, or we think that it was extinct. Yet we haven't even characterized like material from the place that we think it originated. And I think, before we make those conclusions, we really do need to look at this. But anyway, so we were interested and we went and did quite a number of things with that data set. But one of the things that we did with respect to iron and zinc was to do association mapping, obviously. And we were able to find some really interesting QTLs, but the one that I'm highlighting here is the one that was consistent for iron and zinc, explaining up to about 30% phenotypic variation, which is indicative of this being a major QTL. And we did identify some really interesting candidate genes that we are currently characterizing. That same study also did extensive work on agronomic traits, and we do have some really interesting results. So watch out. I mean, this is already accepted in theoretical and applied genetics. So watch out for this paper that's going to come out soon, so you can see some of the other results that Odeny was able to find. But at this point, I'm going to wrap up and just say, with respect to finger millet, we continue to build resources, but I can't say this enough. We really do need to understand the germplasm. We need to understand both the host and the pathogen in terms of with respect to blast disease, but there are also many other diseases, or there's also striga that we do need to understand both the pathogen and the host, and also to fully characterize some of the traits of interest, just so we can prepare this crop for genomic selection. Not just finger millet, but most of the dryland crops remain underutilized, under-researched, but they do have very unique attributes, so they do deserve attention. They're getting a little bit of attention now. We know that VAX, for example, is putting some attention to these crops. But maybe the question we need to ask, are we allocating enough resources to be able to fully exploit what is existing? And especially in this case of finger millet, did we actually allocate enough resources for just conservation? I don't believe so. What can we do to ensure that we don't lose the diversity that exists? But also, I mean, the questions that we need to ask ourselves: can we utilize existing resources to fast track productivity? I think there's almost like some kind of rush to, like, when we get into understanding a crop and an underutilized crop, the first thing we want to rush to do is whole genome sequencing, and especially when PhD students are involved, because it sounds cool. We want to get cool results, but in some cases, I mean, I shared a presentation where we looked at how these reference genomes we've generated are being utilized, and they are highly underutilized. In most cases, we might just need to use the resources that are already existing to fast track productivity of some of these crops. So we also do need to prioritize what we are investing in. I think sometimes whole genome sequencing is overrated, but at the same time, we do need to build capacity, which is what we really try to do, that all our research is done through capacity building. Obviously, strategic partnerships is probably what has brought us here. And I want to acknowledge that I do have quite a number of students and colleagues from my group who are joining. Thank you so much. But we are all quite interested in networking and, looking forward to network in this particular event. I just want to finish by thanking everyone who has contributed either directly or indirectly. It's a lot of work and there are way more people than I could put on this screen. But I just want to thank you all for your contributions and also to thank all the donors and collaborators who've contributed to this work. Thank you so much. And I look forward to any questions. I hope I didn't, I probably just exhausted all my time, but if there's time for some questions, I'll be happy to take it.
Thank you, Dr. Damaris, for that wonderful presentation, which is comprehensive. And I think we can have some minutes for questions. With me, I have received 4 questions. And before you answer, I want to inform participants there is a certificate of attendance at the end of this question you'll be given. That's it. So keep in touch. So, Dr. Damaris, I have a question from Tigles. I hope I pronounced the name well. Tigles. How relevant are consumer acceptance traits, and have you done characterization for them alongside other evaluations?
Yeah, great question. Yes, so there's something called looking at characterizing market segments that has become really important, and especially for crops like finger millet, which, is not so much, it's not broadly known. So we have a group of, we have a network, but we also have a group of scientists working on characterizing the different market segments. And so what we do now is we have target product profiles where we know in a particular country, these are the products that we are looking for, and we have benchmark varieties. So for example, if you're looking for early maturing brown seeded seeds, for a country like Kenya, then you'll have a benchmark variety. Now that variety is what, if you're breeding for that particular segment, it will be your benchmark. But then it goes to different things. It's either for preparing porridge or for breakfast cereals. So that has all been characterized. And in fact, we are operating all the traits that we're looking at, for example, the iron and zinc, these go to different target product profiles. Yeah, so that's a really good question. But yes, we are linked to the consumers and getting feedback. And, we know that these are the kinds of traits that we're working on. Obviously, more needs to be done so that it is better refined. And also when we're doing trait discovery, that it needs to be better refined so that we are really providing specific targets. And the reason I'm saying that is because even though we know that finger millet is high in nutrient content, and nutrient content is one of the things that we need to sell, we don't know the dynamics per country. So we just, like in Ethiopia, we are just beginning to understand. But even the material that we used there is not exhaustive. We don't know if we ended up doing more characterization that we would get something even more. And the same case applies to other countries as well.
Thank you. I hope that one's well answered. The question number 2 is from Femi Patrick. The question is, he starts by saying, brilliant presentation, excellent work by you and your team. Congratulations.
Thank you.
I have a question regarding the marker identification on specific chromosomes. Have you confirmed whether any agronomic traits such as yield are linked to the markers in addition to the disease traits?
Yeah, so I just didn't get to present it here. So for example, the work that was done by Emmanuel, he also looked at agronomic traits. And what we find usually is with higher incidence of blast, you get lower yield, which, I mean, you would expect, but in some cases it's not the same. And this was particularly when you have leaf and finger incidence, not so much with when you have neck and finger blast incidence or severity. It's not so much with the leaf incidence. And so what it indicates is that the most important blast incidences that we need to look at are for finger, let me share this, let me put it on presentation mode, is for finger and neck because they have a penalty on the yield that you end up with. Yeah, that is one. But I think the other example I'm going to give is for nutrient content. The work that Odeny did also, he looked at agronomic traits. Now, there have been theories saying, oh, if you improve the yield of finger millet, then you're likely to reduce the micronutrient content. We didn't find any such correlation. In fact, we found quite a number of very high yielding, but very high nutrient containing genotypes. It's just that we haven't characterized most of these genotypes enough. But there seems not to be any penalty as you increase the finger, either the grain size or yield with micronutrient content, and in this case, specifically iron and zinc.
Thank you. I'm seeing someone raising on hand. Edwin, do you want to ask a question? It's like he's not ready. Let's go to the third question. That question comes from Gobesi from Ethiopia. First, he appreciates that it's a good presentation. Is it really that iron can vary in this much among finger millet genotypes?
Well, science doesn't lie. I mean, we did in 3 environments, and we saw that much variation for sure. It is true. And we didn't work with 100 or 200 genotypes. We worked with 448. So clearly, there is that much variation. And we've been breeding finger millet for like 30 years now, and we still haven't really exploited it, and we talk about it. So what does that tell us? That there's so much more, or maybe our investment is not in the right places, or our prioritization is not in the right places. But clearly there is that much variation for micronutrient content in finger millet. And that was just in one country. We haven't even gone to South Sudan yet, which also has really, really interesting material.
Thank you. The fourth question comes from Prejuice. Could it be that the breeding focus for finger millet in Ethiopia has for long been on agronomic traits rather than the mineral content? Kindly, could you explain could you expand little bit more on the dynamics?
Yeah, I mean, good question. Good question. But in this case, it doesn't matter. So I chose to just present for iron and zinc because I wanted to present something that's relatively unique. But it doesn't, whether we just chose to do agronomic traits only, it doesn't explain why we are not exhausting this germplasm or this germplasm or this germplasm. Why do we have all the released varieties clustering here? But at the same time, when we did the same ranking, so this ranking I indicated here, where we looked at the top 10, we did the same ranking for agronomic traits, yield. Maybe I should have included yield here. There was none of the released varieties that was among the top 10. It was still dominated by Tigrayan material that are not represented in the released varieties. They were the most yielding. In fact, almost every trait that we ranked, the released varieties were they were low ranking. They were very low ranking. And it's not surprising because they are quite highly related and just from one pool here, right? And so, I mean, for as long as we are not exhausting the resources that we have, we don't expect to get the genetic gain. So even if the focus was just on yield, but you're just looking at these 5 individuals who have this narrow range in yield, you're never going to increase your genetic gain. You have to broaden and ensure that you're using the resources that you have within that country. But in this case, you don't even have just that country. You also have a whole region where you can exploit from, and across the region as well. You know, you have the secondary gene pool in India and Nepal.
Thank you. I think we need to take only one more question and then we wrap it because of interest of time. And the last question, we are sorry if we do not answer the other question. Maybe you may write if we get some other time. We'll see how Dr. Damaris can respond to them. But for interest of time, let's finish with this one. But Isa Abdul, thank you very much. Please, during the presentation, you did mention that whole genome sequencing is overrated. And so I want to know what are the methods that are more confirmatory to our target products than whole genome sequencing. Thank you.
So the first objective, as you are going to sequence your genome, you should ask yourself, what is my expected output? What am I looking to achieve? And if what you're looking to achieve is to improve the yield, think about, okay, I want to improve the yield and I want to use advanced tools. What else is there that I can use? And there are cases where it is justified. And I do know, for example, in finger millet, it was justified. We had this tetraploid organism that we couldn't really, it's also very highly self-pollinating. And whenever we tried to develop markers, they were not polymorphic enough. So we needed to understand the genome dynamics. So in some cases, it's justified. But in some cases also, you have very high transferability of markers between, so for example, between rice and finger millet, could we have introduced markers from rice, which is well studied, but also closely related to finger millet? Can we, even right now, as we are trying to characterize the pathogen, is there something that we can transfer from rice to finger millet that we don't have to invest millions of dollars for? So long as you understand your end objective, no problem. But if you are just doing it because, I mean, most of the times you're justifying that it's going to increase your genetic gain, but you haven't exhausted other things that exist. Would you have been better off investing that $1 million? I'm just giving an example, to just characterizing the germplasm first at this level, and exploit what is there, rather than go into whole genome sequencing. I think just align your objectives well with your resources. Yeah.
Thank you, Dr. Damaris, for that informative presentation and answering all the questions. Due to interest of time, we have cut you short and cut the questions from the participants, but we know there's also a way that we can arrange so that they can get the answers for that. We can see how to do that. Now,
yes, I will do my best to respond to all the questions on the chat. Thank you. Thank you all so much.
Okay, thank you very much. Now, at this time, I think I need to hand over this to Dr. Paul to take us to another session. Thank you.